friends, so now I'm back with my next video, and in this video we are going to talk about protein folding. So we have already talked about translation where there is formation of protein, and now we will talk about how a long chain of amino acids, which is a protein, how does it fold in its proper conformation to have a functional role and to act as a protein as an enzymatic activity and other function. So please make sure to like, share and subscribe my channel. So what are proteins? It's a large complex molecule which has many roles, like they play roles in structure, function, regulation of body tissues and organs as well. And the shape into which proteins naturally folds, it's called its native state. The other state can be misfolded protein, but there is one conformation in which the protein exists, and this state is called the native state, which in most cases is determined by its sequence of amino acids. And proteins are mainly, when they are folded, they are held together by different forms of interaction. These interactions are mostly thermodynamically stable, provide the thermodynamic stability to the complex. It can be the hydrophobic interaction, ionic interaction, and disulfide bonds which are formed in the proteins. And there are basically two types of protein based on their structure and function: the fibrous protein and the globular protein. So what are these proteins? So here you can see the globular protein. It's a compact form of the protein, and it is water soluble, which means it contains the hydrophilic side chains which can be soluble in water, and it plays important role for enzymatic and molecular actions. And for the examples could be hemoglobin, myoglobin and insulin, which plays role in different pathways as well. And now, if we talk about the fibrous protein, so you can see the difference. This is a compact structure, but the fibrous protein, it's a long protein filament, and it's mostly water insoluble, which means it contains the hydrophobic side chains which are not soluble in water, and it majorly play role in stability. And the example is collagen or elastins. So there are different structures of protein based on their folding and how they interact with each other, and they are basically primary structure, secondary structure, tertiary and quaternary structure. So basically the primary structure is just the sequence of amino acids. So here you can see a long chain of amino acids where the amino acids are mostly joined by a peptide bond. So here you can see one amino acid, second, third and fourth, in which the two amino acids are joined by this peptide bond of CO and NH. So this is just a long chain of amino acids is called the primary structure. So what is the secondary structure? So here you can see the different structure. It can be either alpha helix and or the beta plated sheets. So what is alpha helix? It's a single stranded molecule which is right stranded, and per spherical turn it contains 3.6 amino acids. And you can see it's just a single strand, and between these strands is hydrogen bonding that connects this structure and then leads to the spiral structure. And so it is intramolecular hydrogen bonding which occurs in the alpha helix. So now if we talk about beta plated structure, so here you can see the two strands of the two beta strands. So it's formed by the two or more beta strands by intermolecular hydrogen bonding, and one beta strand, it's basically 3 to 10 amino acids that combine to form the beta strand. So in this case of beta plated sheets, there can be intermolecular hydrogen bonding, and then happens is beta bands, which is formed by the joining of either two beta strand or alpha strand, and or like the combination of both of them. So this is what is the secondary structure, where different that happens, like the sequence of amino acid, they just the some residues, they just combine with each other in different bondings and leads to what is called the secondary structure of the protein. So what is the tertiary structure? The three-dimensional structure of the protein that we see is basically the tertiary structure, and it is consist of it's a single polypeptide chain with one or more protein structure, and there are different interactions that are involved, like peptide bond, electrostatic interaction, hydrogen bonding, and hydrophobic interaction, disulfide bonds. So here you can see a polypeptide chain with one or more structure which is interacted by either hydrophobic interaction, hydrogen bond, this backbone by peptide bond, disulfide bridge, and ionic bond. So all this interaction is what makes the overall structure of the protein, the three-dimensional structure of the protein, and then helps in the stability of the protein itself. So now that we have a tertiary structure, what comes into play is the quaternary structure, which is mainly formed by the combination of different polypeptide chain. So in the tertiary structure, we have only one polypeptide chain, but in the quaternary structure, we can have multiple folded protein, like one, in this case, 1 2 3 4 structure, which are combined, and they basically combine by either hydrogen bonding, ionic bond, or the salt bridges. So what we have talked about is a primary structure, which is a sequence of amino acids which interact with each other to form either secondary structure, which undergoes further conformation, like different secondary structure come in and what forms the tertiary structure, and then different chains of this tertiary structure forms the quaternary structure, and what we get is the assembled unit which can have a functional role. So then there are how does this protein folds, what factors decide, like now it will go to tertiary structure, and in this way only, like this bond, like it will fold in this way only but not in the another way. So it basically depends, like the major proteins that play a role in the folding are the chaperones. So they are the class of protein that aids in correct folding of the protein in vivo. So what they do is they bind to either unfolded protein or partially folded polypeptide. So they don't actually play a role in, like they don't actually fold the structure, but what they actually do is to prevent the improper folding. So they just bind to the hydrophobic segments to prevent their folding that might lead to protein aggregation and precipitation. And the example is the bacterial Grohl system in case of prokaryotes, or in case of eukaryotes are heat shock protein. So what they do is they bind to either unfolded proteins or the partially folded proteins, and by ATP hydrolysis they prevent their misaggregation and undergo conformational changes, which leads to ATP hydrolysis, and the folded polypeptide is released from this system. So what are the components of the chaperones? So there are basically two components. The first is HSP70 family, which is of 70 kilodalton protein. So what does HSP70 does is it binds to the unfolded polypeptides that are rich in hydrophobic residues. So they binds to the hydrophobic residues to prevent the aggregation, and they, in this way they protect proteins that have been denatured and peptides that are being synthesized or not yet folded. And they also they also block the folding of certain proteins which must remain unfolded until they have been translocated, because suppose there are some proteins which needs to go to other compartment, like from mitochondria to the mitochondria, so they still remain unfolded so that they can cross the membrane easily. And they also facilitate the quaternary assembly of some of the proteins. So we have talked about HSP70, and the another factor is chaperones, which is a large multi-subunit complex. So what in E. coli we are going to talk about is the Grohl and Gross system. So they binds to the unfolded proteins. They binds to the hydrophobic patch, which is present on the open cavity of the Grohl. So the hydrophobic part of the protein, they bind to the hydrophobic binding patch, and then the are basically what I have shown you is, so this is a unfolded or a misfolded protein. It binds to this hydrophobic patch of the binding patch of the Grohl cavity, which leads to the then comes the ATP, and then this is capped by the gross lead, which is in green shape, right? And that this undergo conformation changes leads to ATP hydrolysis, the binding, release of gross, and then the folded protein get released. So like nobody knows how the protein folds so fast, like within 10 seconds, what conformation changes occur, and how the protein actually decide the way to fold. Chaperones just help in the folding, but they don't decide how the protein fold. So there are different theories that people have proposed how the protein folds. So first theory is the framework model. So in this model, the folding starts with the formation of secondary structure. So like suppose formation of secondary structure at one place and the another place, which then these secondary structure interact to form the more advanced folding intermediate. So here you can see one secondary structure which is formed like by rapid formation during protein folding, and this is one of the model, like the framework is to form the secondary structure and then which they interact to form the folding intermediate. So the another model is the collapse model, which is the collapse of the hydrophobic cores. So because the native proteins they contain the hydrophobic cores, so could be the driving force is the in protein folding is the hydrophobic collapse, which means that the hydrophobic residues they come and form interaction, which is a major driving force for protein folding. And what they fold is known as the molten globule, which is a species which merges of a secondary structure but not the tertiary structure. And then in the final folding, the protein undergo complex motion in which it attains like the side packaging, hydrogen bonding, which and it expels water, which then leads to the proper structure. So what it says is the major driving force is the hydrophobic collapse of the like the hydrophobic cores and the formation of the hydrophobic cores by expelling water, proper folding, the complex motion. So next model is the nucleation condensation model. In this model, the secondary structure or the tertiary structure that are made that at a time made. So and the hydrophobic core collapse in a random fashion and forms a native structure. And then the basic like the this secondary structure, tertiary structure, they help in the efficient folding, and they act as a nucleation to add another like folding happens, and they evolve to the efficient folding pathway as well as the native conformation. So these are the three protein folding model that are proposed by different people, and we still don't know how the protein folds so fast and what are the factors deciding like the correct folding. So I hope it's clear. If you have any doubt, please let me know in the comment section, and please make sure to like, share and subscribe my channel. Thank you.